Okay, we're going to take a look at cells, the organelles or the little parts found within the cells. You need to know the difference between prokaryotes and eukaryotes. Those are two types of cells. And then be able to compare and contrast the difference between plant and animal cells. When we take a look at the cell and cell cytology, the cell is the basic unit of structure and function in a living organism. When we look at the order of specialization, you start with the organelles. Again, these are the parts within the cell. They make up the cell. Cells combine together to make tissues. Tissues combine together to make organs. Different organs make our organ system. And then the different organ systems together will make up a multicellular organism. Cytology is the study of the cell. There are two types of cells, prokaryotes and eukaryotes, and you will need to know the difference between them, the specific differences that they have. And then just to give you an idea for their classification, which we will get into in more detail in a later unit, but the prokaryotes consist of the archaea, which is the archaeobacteria, and then the bacteria, which is the eubacteria. Your eukaryotes are the eukarya, which consist of everything else, the protista, fungi, the plantae, and animalia. Prokaryotic cells, your prokaryotes, think pro, no. Pro rhymes with no. No what? No true nucleus. Prokaryotic cells do not have a true nucleus. Prokaryotic cells do not have a nuclear membrane that surrounds the DNA. So where is the DNA? The DNA is found within the cell, but it's not within a nucleus. Prokaryotic cells do not have membrane-bound organelles. So the organelles, the parts inside the cell, do not have a membrane around them. And then prokaryotic cells, these are simple cells. Examples are bacteria, cyanobacteria, and archaeobacteria. Eukaryotic cells, you do have a true distinct nucleus, and then the DNA material would be found inside the nucleus. Eukaryotic cells have nuclear membranes that surround the DNA. Eukaryotic cells also have organelles that have a membrane surrounding them. The eukaryotic cells are definitely more complex. This would include your fungi, protista, plants, and animals. First, let's take a look at comparing an animal cell with a plant cell. Let me grab my pen. You'll notice that the structure between an animal cell and a plant cell is different. In addition to that, you guys need to know the 3 C's. And we call them the 3 C's because it's just a way for us to help remember the 3 main differences between a plant cell and an animal cell. 1, we have the cell wall. Animals do not have cell walls, only plant cells. Then we have a large central vacuole. This only says vacuole, but we tend to say central because it's very large, almost half the cell. And then the 3rd C is the chloroplast. So those are the 3 structures that you would find in a plant cell, but not in an animal cell. Cell wall, large central vacuole, and the chloroplast. Taking a look at the size comparison, we're looking at the sizes in microns. And to give you an idea how small a micron is, it's a millionth of a meter stick. Basically, if you looked at a human hair, you would look at 20 microns for the diameter of one single human hair. So we're talking about something that's only 1 micron, and your human hair is 20 of those. So just to put this in perspective, bacteria range between 1 and 10 microns. A virus, remember viruses are going to attach to that bacteria. Viruses are not living, they're smaller than the bacteria. And then our eukaryotic cells, such as an animal cell, these range between 10 and 100 microns. Okay, looking at the cell and the organelles, the structure and function. Remember, the organelles are the small membrane-bound structures found inside the cells. They're little parts to it or separate compartments. These are the organelles that you need to be familiar with. The cell membrane, which is also known as a plasma membrane or cell surface membrane, depending on which one we're looking at. Cell wall, chloroplast, chromatin, chromosomes, cilia, cytoplasm, rough endoplasmic reticulum, smooth endoplasmic reticulum, the flagella, the Golgi apparatus, also known as Golgi body, lysosomes, microfilaments, microtubules, mitochondria, nuclear membrane, sometimes known as nuclear envelope, nucleolus, nucleus, centrioles, vesicles, ribosomes, and vacuoles. In addition to knowing their functions and structures, you need to be able to identify them within the cell. 3 main parts to a cell. You have the nucleus, the cell membrane or plasma membrane or cell surface membrane, and then the cytoplasm. This is representing the plasma membrane, again known as the cell surface membrane or just cell membrane. There is a plasma membrane found around both plant and animal cells, and it controls what can enter or leave the cell. Looking at the structure of the cell membrane, the cell membrane is a double layer of fat. Specifically, we're looking at the lipids. It's a phospholipid bilayer. Bi means two, so there's 2 layers to the cell membrane. And a phospholipid means you're going to have a phosphate attached to the lipid. That phospholipid is going to consist of your phosphate head. We call this the head, which is hydrophilic polar head, meaning it likes water. And then it contains 2 fatty acid chains, which we call the tails. They are hydrophobic, meaning they do not like water. When you look at the cell membrane, here's the diagram. We said it's a bilayer. Here are the heads and their tails. This is one layer. And then the second layer, here's the heads and then their tails. The tails are pointing towards each other. Remember, the tails are hydrophobic. They do not like water, so we don't find water inside here. The heads like water because they were hydrophilic. These heads are pointing to the outside of the cell, and there's a watery environment found there. These heads are pointing into the cytoplasm. Remember, the cytoplasm was a watery environment. Hydrophilic heads, they like water, hydrophobic tails. Within the cell membrane, we also find proteins. The proteins can be embedded within the membrane. When you look at this bottom diagram, the red kind of like dots are representing the heads, and then inside here, it's kind of hard if you follow my pointer, inside here you're looking at the tails. And then the green structures are the different proteins that are embedded within the cell membrane. And we have receptor molecules, different types of proteins, receptor and transport, and they have different functions within the cell membrane. Looking at the function of the cell membrane, it separates the inside of the cell from the outside environment. Remember, within the cell membrane, we had the two types of proteins. We had transport proteins, which are going to control what enters and leaves the cell. And then we can find the receptor proteins, which communicate between the cells and with anything within the environment. These receptor proteins are going to help maintain a chemical balance in the cell known as homeostasis. Cytoplasm, it's a jelly-like mixture located outside the nucleus. This is where we find our organelles. So the cytoplasm, all of this beige area in here, okay, and it's found throughout the whole cell outside that nucleus in here. That's all representing cytoplasm. And then these structures embedded within the cytoplasm, those represent your different organelles. The cytoplasm is roughly 70% water. Many important chemical reactions occur in the cytoplasm, such as protein synthesis and glycolysis. One of the organelles that we find are the centrioles. The centrioles are used in cell division and replication. We'll get into cell division and replication more when we talk about the cell cycle, and then specifically look more at the function for the centrioles when we get into mitosis. Vacuoles, they transport and store food and water. Vacuoles can be used to temporarily store enzymes or other materials needed by the cell. If needed, they can even store waste. Remember, plants have that large central vacuole that's full of water to help provide support. The vacuoles found in animal cells are much smaller. Lysosomes, they are membrane sacs. They're filled with digestive enzymes that break down excess or worn out cell parts, food particles, or invading viruses or bacteria. They can fuse with the vacuole, dispense their enzymes into the vacuole, and then digest food. We kind of think of lysosomes like Lysol lysosomes. They're like the cleanup crew. They like to get rid of all of the garbage and the recycled material. Sometimes cells need to die. Lysosomes can be used to kill cells when they are supposed to be destroyed. Some cells need to die as part of normal development in an organism. It's an auto-destruct process where the lysosomes break open and kill the cell, kind of like a cell suicide. An example of that would be a tadpole becoming a frog. The tadpoles have to reabsorb their tails in order to become a frog. Vesicles function as transport materials throughout the cell, kind of like a shipping box. They are small spheres of membrane. It's the same material that you would find in the rough endoplasmic reticulum, the Golgi apparatus, or plasma membrane. It's part of an endomembrane system where it can fuse or pinch off from other membrane-bound organelles, and they're found in all eukaryotic cells. If you look at this diagram at the top, they're showing you where you have a little vesicle, and it actually fuses. Fusing here becomes part of it so that it can open up, and whatever was inside its little shipping box, if you will, inside that vesicle can be released on the other side. Looking at the mitochondria, some call it the mighty mitochondria. Mitochondria has a double membrane. Some organelles have double membranes, some organelles only have one, but mitochondria has a double membrane. It's folded in layers to give it more surface area so it can make more energy for the cell. The mitochondria are the sites for aerobic respiration. They have enzymes that break down food molecules to release energy. The cristae, and if you look at the diagram here, the cristae, this folded membrane inside, that's a folded membrane known as a cristae that provides for the larger surface area for these reactions to occur. Mitochondria is responsible for making ATP, which is going to give us our energy, and it performs cellular respiration. And we will go through this process in more detail later, but basically sugar and oxygen can combine in a chemical process producing the energy, and this is what's going to give us the fuel for life. Mitochondria are found in both plant and animal cells. Plants make energy in two ways. One we just mentioned was mitochondria, where they make energy from a chemical reaction involving sugar and oxygen in a process known as cellular respiration, producing, notice your ATP is a product here. And then a second way is from chloroplast, where they can make energy from sugar from the sunlight in a process known as photosynthesis. So ATP is the active form of energy, while sugar is a stored form of energy. The nucleus is the control center of the cell. Within the nucleus, we can find chromatin, which is the long thread-like structures, kind of looks like a plate of spaghetti, if you will, is found during normal periods of the cell activity. The chromosomes are when the chromatin starts to coil up and gets tighter and tighter to make what we call that X-shaped chromosome, and that happens during cell division. The nucleus is going to protect the DNA. Remember, in bacteria, there isn't a nucleus, so the DNA is just found within the bacteria cell. The nucleus has a nuclear membrane, sometimes again known as a nuclear envelope, that's going to encase the whole nucleus. It's going to separate the nucleoplasm from the cytoplasm. It regulates the flow of material that goes in and out of the nucleus. Inside the nucleus, you can also find a nucleolus. If you look at the diagram here, this is representing a nucleus, and then this little ball structure is known as the nucleolus. Nucleolus is the ribosome factory, meaning the nucleolus is responsible for producing ribosomes. And then you can also find the chromosomes, or the DNA material, within the nucleus. Chromatin, again, remember I said it kind of looks like a plate of spaghetti? The chromatin, these are strands of DNA material, and it stays curled up like noodles in the nucleus until cell division occurs. It does eventually form that X-shaped chromosome as we are going into cell division. And this diagram is showing you, here's your chromatin inside the nucleus. This purple out layer here is representing your nuclear envelope. There's a nuclear pore where things can pass back and forth. But as the chromatin starts to condense and coil, it'll start condensing and coiling. It wraps around those histones, which are the proteins, and then as it's continuing to wrap around, eventually you start to see that double helix shape, and then it continues to wrap around, and then that's eventually when we'll get our condensed chromosome, which is the X shape that we're familiar with for the structure. Okay, the cytoskeleton, there's two parts to the cytoskeleton, the microtubes and then the microfilaments. The cytoskeleton provides support and shape for the cell. The microtubules are long tubes that give the cell shape and act as tracks for the movement of the organelles. And then the microfilaments are threads that allow the cell to move and divide. Ribosomes are responsible for producing proteins. They're going to read the instructions to build the proteins from the DNA. For the structure of ribosomes, there's two subunits. You have a larger subunit and then a smaller subunit on the bottom. We can find the ribosomes free within the cytoplasm, and then some of the ribosomes are going to be attached to the endoplasmic reticulum. The endoplasmic reticulum, that's part of the protein factory, again, help making the proteins. And then they're also helping with making the membranes. When you're looking at endoplasmic reticulum, there's a rough and smooth endoplasmic reticulum. The rough ER or rough endoplasmic reticulum has ribosomes attached to it. And that's where it's getting the rough name from having little structures attached. The rough endoplasmic reticulum is responsible for helping with protein production. And then the smooth endoplasmic reticulum, there are no little ribosomes attached. The smooth ER is responsible for making membranes. The Golgi apparatus kind of functions as our UPS headquarters. This is where things finish up, it gets sorted out, kind of puts a label on it and gets shipped out to the areas that it needs to within the cell. The proteins, you have that membrane-bound structure with the little carrying proteins or vesicles attached, and those little proteins are going to act as UPS trucks where they've got their information packed in them and they can go and deliver that to the rest of the cell. The flagella and cilia. Flagella are long whip-like tails that allow the cell to be moved along. And then the cilia are small little hair-like projections that kind of flow back and forth, again helping with the movement of the cells. Just looking at another diagram of an animal cell with the different organelles labeled. And then we have our plant cell again with the organelles labeled. Remember, the plant cell has the 3 C's, the 3 organelles that are found in plant cells and not in animal cells. Those 3 C's were your cell wall, chloroplast, and the large central vacuole. The cell wall contains cellulose, which is one of our polysaccharides. Cellulose is hard to digest. It adds fiber to the diet, and it makes up the primary wall of the cell wall. The cell wall is to provide the plant with its shape and its support. The cell wall surrounds the plasma membrane. So if you look at the diagram of our cell, here's the cell wall on the very outside layer, and then just inside that is where we find the cell membrane. Chloroplast is the site for photosynthesis. This is where plants can transform sunlight energy into glucose. Plants contain a green pigment called chlorophyll that is going to help trap the sun's energy. We will go into photosynthesis and the chloroplast in more detail. And then the third, the large central vacuole, which stores water and regulates something known as turgor pressure. Turgor is the pressure exerted by fluid in the cell that presses the cell membrane against the cell wall. In the bottom picture down here, representing the plant that has a high turgor pressure, where you see your plant looks very happy there. The second diagram where sugar pressure is low, it's called a flaccid state, meaning the plant needs water in order to build that pressure back up. And if you look at the cell diagram, turgor pressure, the large central vacuole is nice and full of water. When you have a low turgor pressure, that central vacuole is much, much smaller. Okay, one last point. We're looking at cells and the organelles found within the cells. We need to also understand that the cells have to be able to reproduce. We need to have old, worn out cells replaced. Cells that have been damaged need to be able to be replaced. And then for growth, we need new cells. Binary fission and mitotic cell division are the two processes in which a cell reproduces through something known as asexual reproduction. Asexual reproduction just means it's starting with one cell. And if you look at this diagram, binary fission begins with one cell, and then mitosis also begins with one cell. In the end, after the reproduction, binary fission ends in two cells and mitosis ends in two cells, and there's processes that occur along the way. In binary fission, the cell is literally going to divide in half, and you see here where the cytokinesis has occurred, dividing the cytoplasm. It literally splits the cell into two. In mitosis, there's a process that this cell goes through. That process is prophase, metaphase, anaphase, and telophase. And we'll go through mitosis in much more detail at a later time. But for right now, understand that binary fission, this is typically for your single-celled organisms. Your prokaryotes go through binary fission. They reproduce by dividing in half, splitting. And then all of our eukaryotes are going to go through mitosis, in which that's going to involve part of the cell cycle where we have prophase, metaphase, anaphase, and telophase.